My name is Higgs and this is my friend Boson. Welcome to the Higgs world.
Thanks for the introduction, Higgs. In this presentation, our goal is to offer you a quick summary of the research and conclusions. We follow inquiry-based learning model where students take lead in learning.
First, they get a quick review of the research via the videos. Second, they review the paper to learn and understand key concepts. Finally, they can follow up with further research on their own to dig deeper. In the end, students would have learned something new, latest and relevant. Isn't that wonderful, Boson?
Great point, Higgs. Please follow the links to review the research in further details. We want to thank all the authors and publishers for making this research public, thereby advancing science and innovation. Please like, share and subscribe.
Today, we are reviewing a paper titled, Formulation of New Media from Dairy and Brewery Wastes for a Sustainable Production of DHA-Rich Oil by Aurantiochytrium mangrovei. The dairy industry is one of the main food industries in Italy and Europe, with tons of cheese produced every year. In Italy, the main dairy product is the mozzarella cheese, with a production of more than 250,000 tons every year. During the mozzarella manufacturing process, two main side streams are generated: the cheese whey and the mozzarella stretching water. Mozzarella stretching water is treated as an effluent by the dairy companies because of its high salinity and high chemical oxygen demand, and therefore it represents a serious environmental issue.
Another promising food waste for biotechnological applications is the spent brewery yeast. It is an organic waste from brew manufacturing with a high content of proteins, free amino nitrogen, phosphates and other essential mineral salts. This food waste produced after brewery manufacturing that could be recycled in new biotechnological processes.
That is so intriguing, Boson. The purpose of this study was to investigate the potential of a dairy wastewater, in combination with spent brewery yeast, as a new sustainable growth medium.
Aurantiochytrium is a genus, industrially exploited for the production of docosahexaenoic acid. Aurantiochytrium can produce high amounts of lipids up to 55% of dry weight, and most of that is docosahexaenoic acid. Docosahexaenoic acid is an important fatty acid, as it is one of the major components of the central nervous system. This makes this paper very relevant.
Great point, Boson. The results are very promising. The optimization with response surface methodology leads to a biomass dry cell weight of 10.14 grams per liter, with 38.9% of lipids and 29.8% of docosahexaenoic acid, on total fatty acid methyl esters. The results are comparable to the relative growth, with standard media.
That is wonderful, Higgs. These findings suggest that hydrolyzed mozzarella stretching water with spent brewery yeast can be used in new biotechnological processes, in order to reduce nutrient costs for production of biomass that is rich in docosahexaenoic acid oil. In conclusion, the combination of wastewater from mozzarella manufacturing and brewery waste showed a promising alternative for a more sustainable Aurantiochytrium mangrovei cultivation.
Pre-treatment of mozzarella stretching water is mandatory to achieve an optimal biomass concentration and lipid production. That was an interesting paper. Hope you liked it, Boson.